welcome to our April coffee chat. I am Laura Knutson-Murray. I am the GIS coordinator and community outreach assistant with IrisBG. With us today is Shanna Jones. She is our social media and community outreach assistant, and also Mari Rustin, director of strategy and product manager, and Oyvind Rustin, the founder and CEO of IrisBG. You are able to follow our coffee chats in our forum. Just click on it and you will be notified via email when a new article is published. These coffee chats are for you. If there's something that you would like us to give input on or a topic you would like us to address, please let us know. We're able to give demonstrations of the functionality of IrisBG, or if you have feedback for how it could improve, we're all ears. We don't have a fixed format for these sessions, so if there's anything you'd like us to present on, a challenge, big or small, just let us know. Hopefully you will make some new connections today, and together we might become a little bit wiser. Save the date for our May coffee chat on May 14th. We will be hearing from James Miller from the Sheffield Botanic Garden, and he will cover the Garden Explorer. And just a quick heads up, if you are in the UK or able to travel to the Royal Botanic Garden Edinburgh, there will be a free training given by Shanna Jones, 2 sessions. The first one will be for all roles and types covering permissions, and the second one for IrisBG admins, botanists, and curators going in depth. And afterwards, there will be a behind-the-scenes garden tour. So you can scan the QR code, and we have a registration form. You can see more details. And for those of you who are in the States and thinking about going to the APGA conference this year in Boston, just down the road, we will have a Coffee Chat Live, our very first Coffee Chat Live, where we will have cover training. We have new staff, and we'll talk about preparing workflows for entering data. And we'll also have a hands-on workshop that will cover how to use the IrisBG ArcGIS. Sign up for that. And just a quick map to show just how close it is. It's a 12-minute car or train ride, or if you're up for it, a 32-minute walk. So it's very close by. One day early, it's on Sunday, and we hope to see you there. Here is just a look at everyone who registered for the coffee chat today. It's fun to see so many of you from all over. Took time out of your busy spring schedule to learn about propagation, so thank you for coming. Let's launch our poll. We have our usual poll where you can rate your experience using IrisBG. Are you a novice, intermediate, advanced, or are you just trying out the trial? 76% of you are intermediate, 19% advanced. And then the next poll, just to give our speakers some context for how many of you are entering propagation data. Just answer yes or no. Are you using propagation records with IrisBG? Okay, it looks like about half and half. 55% said yes, 45% said no. Great. Our agenda today, we are so excited to have Peter Brownless and Ross Irvine from Royal Botanic Garden Edinburgh to share their propagation protocol that they use. And just a quick reminder, we had some of the best nursery managers in the world come together this past summer to share their configurations, codes, workflows, and we would have loved to have all of them share with you, but because of our time limit, we just have one of them sharing this time. But there is a knowledge library article that covers just case studies of how those different gardens approach propagation that's shared in our knowledge library. It's more than one way to configure Iris, as we all know, and I'll hand it over to Ross and Peter.
There we go. Well, as a starter picture here, I've got a picture of our second site. We're actually now on a third site, and this is what our garden looked at. And the building that's at the front there, we moved relatively recently from its site to our third site now, and it now serves as part of our community education. So I'm going to give a little bit of context to it. Why didn't that move on? So to give a bit of context, I've got an aerial view of the garden here. We were formed in 1670, and as I said, we're now on our third site. And to give some context for the American audience, we're a kind of zone 9, but we're never very hot or never very cold. So that's the kind of temperature range we work in. And the main garden here in Edinburgh is about 70 acres or 28 hectares. And we've been running records basically since almost day one, and we have almost all of the record keeping kept in our library. So from about 1670, we have some beautiful handwritten accession books, some of which are incredibly difficult to read. And then in the 1950s, we actually moved from accession books to a card index system, which makes it slightly easier to find some of the information that we've got. And then we started to computerize, actually very luckily, in the 1970s with part of the Scottish government's computer system. And then in 89, we moved to BGBase, and we're a relatively new convert to Iris. We moved in 2019. Record keeping here is a bit of a challenge because not only are we on one main garden site, but we also have 3 satellite gardens, and we also manage data for, I think, somewhere in the region of now 50 sites within the UK, contain conifer conservation. We have an alpine outstation, and we have a number of sites that we monitor for native plants within Scotland as well. Our main collection, as you can see, is really quite large. We have over 40,000 living plants recorded in the system, covering well over 11,000 main taxa. And the claim is that we represent 4.75% of all known plants, which gives you some idea of the scale of the collection and the challenge that I face. So here are some of the early accession books, which contain all kinds of hidden gems, like letters from people that sent material to the garden. And behind, you can see the catalogue of plants as was, which was basically the card index sitting underneath. The early precious accession books are kept in a different, separate place. And then this was our first computer, which, being getting on in age a bit, I can actually remember this sitting down in the basement of our herbarium. It made a terrific racket as it took the information off and on the discs that went in those enormous slots. And actually, if you look at the prices, it was an awful lot of money in those days. And then some of you may be familiar with BGBase, but most of my record keeping life here has been based on using that. So currently we have a little bit of a split system. When we used BGBase, both our herbarium and our living collections were on that, but the herbarium found that wasn't sophisticated enough for using for the many various different names that they like to call plants. So they moved to a system called Specify, and the living collection moved itself to Iris and Floria. What we are trying to do here with our record system is to try and tell a story so that when people look at the data or extract data from Iris, it basically tells a story from the collection of the material, collecting as much information about the plant material as is at all possible, how we acquired it, did it come to us direct from the wild, did it come to us through a second person? And it's predominantly myself and my colleague Ross and another chap who is on the call called Nate, who do most of the accessioning for the garden. We then in the United Kingdom have a lot of plant health regulations, so all material that comes into the garden, if it comes from outside the UK, enters the garden through quarantine, or it enters the garden from inside the UK through isolation areas to try and keep out as much unpleasant diseases as possible. Then in most instances, the data then starts its life in the propagation file, and eventually, as Ross is going to outline, finds itself out into the living collection. Though we do have a substantial number of plants that I am trying to keep from getting into the living collection, and I vouch for those. Our botanists do like collecting rather invasive daisies from around the world, some of which I really don't want spreading throughout the garden. So we usually try and get those to find themselves onto a voucher rather than a living plant. We then have plants in the garden and we go through a process of reaccession, recording the parents so that we can then keep a track of the generations of those plants and trace them back to the original plant material. Most of our propagation is vegetative for material that we already have within the garden because we're trying to preserve exactly what was collected in the wild. And in certain instances, it may be not possible to keep the plant material going. And through the process, we also want to learn as much information about the plant material as possible. So frequently it's vouchered or a DNA sample might be made. And all of this is basically to get us to something that we can publish so that people actually see the work that's going on. This is an example of publications that have come out of RBGE. We have our own in-house magazine to share as much of the data as possible, and for specific things like one that I've been involved with, threatened plants of Chile, which has a huge amount of propagation information built into it, and all of that is taken out of a database so that you almost immediately, if somebody asks, how do you propagate something, if you look at the taxon records for that, you'll find through the accessions the numbers of times that we've tried something and the number of times that we've failed and the number of times that we've also been successful as well. So it gives you some idea of basically how to propagate things, which can be very useful for public inquiries and all manner of other things. So this gives you some example, and this was one from, I think, the last year or the year before, of the amount of reaccessioning that goes on within the garden. So we've coded the plants that come into the garden with the letter E if they come here for Edinburgh, or a Y or a V or a Z if they go to any of our outstations. Then we also try to categorise plants that are relatively new coming in as a reaccession of that as well. We've also got quite a lot of DNA bank material in there, and we recently added our cultivated vouchers into Iris as well because they sit better in there than they do in the specification database. One of the things that we do on reaccessioning as well is add all of the paperwork through permits for things like CBD, so that we know what we can do with the material. So you'll see there under the accession restriction column that quite a lot of plant material has no restriction whatsoever, but there are a few there with red that basically it's going to be a material that's used at RBGE and we actually can't let it go anywhere else without some specific terms and conditions. Increasingly, when we get material that comes in as live plant material, we have to cover things like plant breeders' rights and many other things. So having this record at the early stage allows us to know what we can do with it, and in some cases whether we can actually even propagate it as well. Because we have so many plants, we do many different things. So the material types that we have got into quite a large list these days, and Ross is going to talk in further details about that. But we have tropical greenhouses, we have temperate greenhouses, we have all the plants that will grow outside in Zone 9. And just the sheer number of plants means that we need very many different techniques to actually categorise what we do. We also keep a limited germplasm store. We have that separated into two parts. The original wild collected seed, which stays under one accession number, and a reaccessed set of seed that may have been collected from open pollination in the garden. A number of people have asked us why we don't do an index eminent, and that's the reason why, because all of our garden collected seed is actually from open pollination, so it may not be exactly what it started off as, because we've got so many different plants that could intrahybridize. Working through the basic life cycle of all of these things, it does then mean that you need a whole series of item statuses. And we started off also using some of the statuses that were pre-built in with Iris when we first adopted it, but we've actually tried to reduce them. And actually, it's become an ever-evolving thing. Like, this doesn't meet the scenario that I want to record. Can we add a new code? So the list is growing, particularly about how we actually murder plants in particular. People do like, and it is very useful to record that as well. So you can see there some of the statuses that we've developed relatively recently for our particular collection. As I mentioned, one of the important things for us as a botanic garden is to actually keep as much plant disease out as possible. And we've got, as I mentioned, our own quarantine house for material from outside the UK, and our isolation houses as well. You can see the isolation house down in the middle over here in the nursery, which actually is just behind me on the other side of the shed there. And a typical plant label for commercial material coming in, you can see things like plant passports, which used to be very important for Europe, but actually now we've left Europe, we have our own system of that. So we also record all of that information as well. And all of that I manage in a task so that I can keep track of when material came in, when I look at it, if it develops any pests and diseases through the process. And actually quite recently it's worked very well to keep golden root mealybug out of our collection. We also make extensive use of Floria. Many of the areas within the garden have computers sitting on potting benches, but a lot of the staff that work in the outside areas have access to Floria and do direct input, which we put in every day. This is basically the Floria view of the isolation task, and you can see the notes there that I've made about when the plants were inspected. And this is where I think I hand over.
I didn't really get involved with Iris until actually COVID started, and we got a chance to sit basically at home for several weeks and work on propagation records. And it was a bit of a nervous time. We at the time had, I think, around 63,000 propagation records. About 6,000 of them were still active at the time. So moving everything from one place to the other, from our old database to Iris was quite nerve-wracking for us. We had to do quite a lot of stock taking and moving stuff around, and then restock taking after it all moved across. I think it generally went really well. There's a few things weren't great, but we've managed over the years between myself and Peter and the nursery team to get them sorted. But one of the strange things that we have here is we have a lot of users. Virtually every gardener at the botanics here and the outstations has access to Iris, is able to edit in Iris and stuff as well. This has been great for getting information in very quickly and more information rather than it kind of traveling around in sheets of papers. I know some people are other forms of information exchange. So that's worked really well for us, but it also can cause its own problems. So while I was off with COVID, we actually kind of wrote a book specifically for RBGE, and part of that was how we propagate here, how we record propagations here at RBGE. So one of our major kind of changes was we decided to start, due to kind of environmental impact, the disease impact, we have a very important, one of the most important rhododendron collections, I think, in the world, and we were having issues with propagating it because we're getting such poor growth and things of it, just due to dry springs, and you don't expect that in Scotland, but we do now. And so we decided to start micropropagating, and to trace things properly, we decided to start reaccessing vegetative propagation. One, because we now work on a genetic scale these days within horticulture, so genetics aren't 100% when you take from one plant to another. Unlike flora formulas, which didn't really change very much, which people remember them from the past. So we found our old database was lacking in the ability to do this reaccessing. So it was only the nursery and we just reaccessed ourselves. No one else ever took it up. Searching on reaccessions and things was actually quite difficult. And one of the biggest advantages of Iris, and one of the reasons that really cheered me up when I first saw it, was things like the parentage tab, which we have taken great advantage of over the years, and the item reference tab. So all seed propagation has always been reaccessed. Now all vegetative propagation is reaccessed. Unless there's a specific reason for not reaccessing it, we just reaccess it all these days. So we can trace it all now through the item reference. Somebody coming in to have a look now can actually see, I think, a better story. So this is why we decided to do it all. At the same time, when we first got Iris, Peter sort of made a micropropagation module based on just a normal propagation module, but it separated off the micropropagation and let us use different statuses and stuff that were specific to micropropagation, which you can see here on the screen, and areas that were specific to micropropagation. So that was really good as well. Within our old database, we had to use project codes, which helped us report on it, but it wasn't a great system. So this is much better. This is only for the people who are working on micropropagation, and they can look up their own stuff and find out. Hopefully in the future, our micropropagation kind of felt a bit because of COVID, and we weren't allowed to go near science areas for about a year, and we lost everything effectively. So we had to restart again, and we've restarted with Iris, and this is looking at making it a lot easier for us to report on things that have been success and things that have been a failure. We also do a bit of micropropagation. We also do native orchids as well. So we've been doing them from seed, again, using the micropropagation module. The reason that I like this picture is because actually you can see this was started in our old database. And if you actually look at item number B at the bottom, if you read up, it's actually a bit of a mess. Apparently it was potted up twice before it germinated and things like this. So it's kind of moved a bit all over the place, and then it kind of sorts itself out a bit when we moved into Iris. So seed propagation is one of our main ways. Virtually all of our seed is that we sow is wild collected seed. So it's just brought in and accessed. So, and we do everything through you can see this is actually a mouse-free, well, hopefully mouse-free frame. Unfortunately, it wasn't cat or fox-free, so we had to put a lid on it. And this is, we grow, I mean, this is hardly propagation we do in the nursery here. So we grow everything outside to get a nice stratification period to get the seed germinated. Everything, and we do label everything. Everything in a pot is considered a propagation in the nursery, and it gets a barcode label. As you can see, these barcode labels here. The main picture's a non-barcode label, so it doesn't actually have the barcode. But the ones sticking out of the pots, they all have barcodes on them, which helps us if we use them with Floria and stuff in the future. But even if the barcodes weren't handy, this is still a great system because it means that we don't have to try and interpret somebody's writing, which is what we used to do in the past. Or I found this amazing thing when you read people can get two numbers mixed up very, very easily without realizing it for a long time. Now and again, you had to go and search for something because the numbers were mixed up, and that's what it mostly was just two numbers the wrong way around. So we barcode label everything. We don't need to worry about all that. It's printed straight off. And it means we can track everything with Floria if we choose to. I still tend to do things at the bench, or at the computer, but that's because they're all, all our work is right next to these computers. But that's our barcode label printer that we use to print off all our barcodes there as well. So this was one of the early records that we did. You could see it was started in 2020. So this record has never come from the old database, and actually, there looks to be quite a lot there for such a short period on the kind of story that's told about starting off in E49. The seed's been sown. It was checked at one point about 9 months later, and then it germinated a month after that. It was brought into house. One of the things that's happened over the nursery is we've had a lot of work going on due to a big project happening in the garden. So we've had to swap and move things around the houses quite a bit, and we had to turn some of our houses into tropical houses, and we had to move stuff out. So you can actually see it's moved around quite a lot. And the funny thing is, unbeknownst to us when we made this slideshow, this plant actually left the nursery today to go to be planted in the garden. So we'd had it here, we sowed it in 2020, and it just left today to go and be planted. About 30 plants just left. So this is a good story. This is what I keep saying to people whenever I'm teaching them about Iris, is you have to be able to read a story and what's happened to the plant. So you can see, I always say that, you know, what you want to produce is something that somebody could read in 100 years' time and still understand exactly the story of the plant. One of the other sort of major things we do, Peter mentioned the fact that we keep our own seed. We don't collect too much seed from the garden. We do with things like Echinopsis and Primula, short-lived perennials, purely so that we can keep them going, as we don't collect enough. And certainly we haven't really done any collection in the last 4 years. As we had a trip out to Japan last year and a trip to Taiwan last year as well. And that was the first 2 trips for about 4 years. So we do store a lot of wild collected seed, and we do store a lot of tropical seed. So this is actually a picture of the fridges that we have over the other side. And these are, well, there's begonia I can see on the label there on the left-hand side. So we keep our tropical and our spores. That would also be suitable for a lot of orchid seed storage as well, but I'm not sure if we don't have much orchid seed at the moment. That's something we're going to be working on for Microprop. And then, so we do have germplasm records as well, and we do record everything from, like, there's some fern spores, and the one at the bottom is the Coco de Mer, which is the largest seed in the world, which is about 2 foot across, things, and everything in between. So we use Iris for all these things as well. This is a project that's come up on as recently. We found really useful. We got some money from effectively Nature Scotland to save 10 species of plants in Scotland that are in dangerous levels at the moment. What you see here is the elm tree. We've lost a huge percentage of our elm trees in this country in the last 50 years. But we do have areas where some elms actually survive Dutch elm disease. And so we're pollinating these, and we're sowing the seed and then taking the seed back out and planting it again. And then, unfortunately, we can't really tell. We're hoping it'll survive because the parents have been Dutch elm resistant. We won't really know because it takes quite a few years for an elm to get big enough to die of Dutch elm disease. It doesn't affect the children too much, young trees too much, but it affects adult trees. And because this is a project that has been funded by an outside body, one of the things they require quite frequently is reports about what we're achieving, what we're growing, what we're doing, and all that. So with all this information is stored within Iris and pulled through Iris, using reports pulled out of Iris every month. They have to report on how well they're getting on with everything. So the people doing this have really benefited from the report system and stuff within Iris to get things moving.
And some of the ones that you can see in the pollination control bags in the centre there are actually being planted out this spring.
Yeah, the people doing the project are away this week up to up north planting elms right now. So that's been really good. That's the first of the stuff starting to get planted out. Vegetative propagation. Most of our vegetative propagation over the years was fairly simple, cuttings and stuff. But as I say, I was saying earlier about the environment affecting rhododendrons. It's also affected a lot of our other plants as well. And we have a lot of trees and things that there's no other real way of propagating them other than grafting. You can try hardwood cuttings and stuff and things, and they don't always work. And so, but we always hit things in a lot of different ways. And this is where we separate everything off into item numbers, because we may do cuttings, we may also do grafting of the same plant. We may collect seed from the same plant. There's quite a few of the sorbuses that we try and hit in many different ways just to try and get these plants growing. Grafting's not great for us because that's effectively 2 plants, and we would prefer everything on its own roots for obvious reasons. But the picture in the bottom middle there is a project that one of my colleagues has been working on with lime trees. We had the gentleman who had a large lime collection. He wrote the book on lime trees, Donald Piggott. He unfortunately passed away, and his wife kind of wanted us to continue his collection, but you can't go down to someone's house and dig out 100-foot lime trees. So we're grafting all these lime trees. Hopefully we can then French layer or whatever, these lime trees onto their own roots over the coming years, and then get them into the garden. So that'll all be tracked obviously in Iris and stuff. We use tasks. We ask people to fill in tasks to bring in for us to decide what we're going to propagate and stuff. And this was Peter picked this task. It was quite interesting. This is for our herbology course. We have a small herbology course we run here at the Botanics, but this is a propagation request. But if you look at it and you're a propagator, you'll realize that, that everything in here is propagated in different ways. So they send us the task and we decide how we're going to propagate it. I'd quite like them to use the comments to tell us how many plants they want. That would have been quite useful. But of the Bellis perennis, we actually bought in seeds. The daisy, myrica, we can layer it, we can drop layer it, we can sow seed if we get some seed and stuff off it and things. The vincas we can kind of grow on and divide up. So this is how we hopefully get information in, and then we work on the information and let them know when the stuff has been propagated. And obviously they can go back into their own task, click on the accession number and go back and check themselves as to what status the propagation may be at. And one of the other, our old database could tell us the bed of where things were when we go out into the garden or into the outstations to find stuff. But obviously one of the advantages of Iris is the kind of GPS system. And here we created to find some rhododendrons probably for microprop. And then, but we have, myself and Peter were discussing about how many beds we actually have in the garden and the 3 outstations. And I can't imagine it's much below 1,000, to be honest. So, but we can go to these beds and we can actually see whereabouts in the bed the plant roughly should be. We can also switch on the GPS and it'll show you whereabouts you are as well, so that you know how close your little red marker as you walk towards the green marker. They hopefully get closer and we can find the plants much easier. This is simplified finding plants. We don't have to. I mean, we have in some areas we have a lot of rhododendrons planted together, and you're going through each individual one. Is that the one I want? No. Is that the one I want? Whereas this helps really well. We don't do too much tropical propagation or anything, but it is all traced and things from leaf cuttings. The one thing that I think is not great, and I've always kind of said that when you get a free hand at putting the information in yourself, after a few years it can get a bit dangerous. And the treatment, one of my best examples is the treatment box there, which I quite like to use. But the old database treatments were pulled into Iris, and we must have hundreds of treatments. And I don't know how many times you can misspell closed case, but it must be spelled, if you scroll down through it, there must be about 20 different spellings of closed case, as one example. So it's become kind of too cumbersome and difficult for us to use. It's one of these things where you give people a free hand to write whatever they want. They type something in, hit return, and that's it for life, added to the system. Failure's not much better, to be honest, and medium's better, but it's still got an awful lot of stuff in it. I mean, I don't know how many times you can describe a 2-litre pot. So these I find difficult to use, to be honest. I do use medium and environment a lot, even those, but I've just given up on treatment, as I find it's just sort of, there's so many, it's just so full of, because I mean, effectively we have 30 years of people putting stuff in it from our old database. And this is one of the other disadvantages of having so many users, is the fact that people can put things in there and you can't really do anything about it. I prefer drop-down boxes where you have a choice, as long as the choice isn't subjective, because people's opinions aren't always the best. This is one of the kind of divisions we've done. This is for, again, the rare native Scottish project, Polygonatum, that we've been dividing. We're trying to get about 1,000 plants, so we're dividing it up and growing it on, and dividing it up and growing it on. And we also want to then, once it flowers, is to hand pollinate the flowers, collect the seed, and grow that seed, and then that'll all be stored again within Iris and traced, and again be able to report upon it to the people who are helping to fund this project and stuff. So it's quite a lot of work going on there. And you can see we never delete a propagation module. You can see there item number B has a red dot next to it, which means it's a completed record. So we're finished with that record. But the C and D are probably propagations from that original record. But we can use item reference to reference back to that if we can look at it. You can see item D references back to the completed propagation. We got, out of the 12 trays that we had, we got 250 trays when we divided it up. So people can tell that these things are going on. And we also, we can travel through the parentage as well over the years. Explaining about the lime project, growing tilia trees thing, but we've had to start using a lot of grafting and stuff now. It wasn't something that I went on a course to learn about grafting. It wasn't something we ever did because, I say, botanically, it's not great for us. But we have had to rescue trees where we have to actually graft and stuff. And again, this is all tracked and stuff, and this is quite good. Yeah, and we can store things like the root stocks and things as well in the little comments boxes, so that we know exactly what the parent gel is. Most of the grafting we do with the aim of eventually getting the plant onto its own roots. We did say that there's a plant that was Sorbus pseudomonicae, is now Hedlundia pseudomonicae, which we know of one plant in the world. Myself and Peter, back about 2010, budded it onto some other sorbuses, and we now have about 10, 15 plants in the nursery, and we have quite a few plants that have went back out into the wild. So now, rather than being one plant in the wild, there's quite a few, and we every year we try and to propagate the plants that we still have in the nursery from that original one plant collection that we did in 2010, and that's all traced and tracked through the database. Once the plants are kind of getting ready to leave, I encourage people to take them by sending them emails, letting them know that we have plants ready to go out and things. Generally in October, we can plant out here really kind of safely before it gets too dry, sort of October to March, I always say, but people will plant out in April and things as well. We don't have a great watering system around Edinburgh, certainly around Edinburgh, automatic system. There is bits and bobs, but so they've got to be quite careful. And once we actually create our own labels here in the nursery, so we do the barcode labels for the propagation, and then we create these engraved labels as well, using using basically pulling out Excel reports out of Iris and then loading it into Lightburn to then use to engrave. So we create all our own labels and stuff nowadays as well. I certainly create a few thousand labels a year for the garden and with the label machine. So as plants leave the nursery, we actually inspect them. Again, we do this through the task field. Myself and Peter and some other people across the road who do plants across the road, we inspect every plant before we release it from the nursery, and then that's registered and kept. I know the tasks are completed, but they're still there to be looked at in the future, and then inspected. And anything that leaves Edinburgh then has to have a plant passport, which we then create and keep within Iris and stuff as well. So there's lots of work to actually, even from taking the seed in to getting the plant out, or taking the cuttings to getting the plant out, that we have to cover. Now Peter's going to talk a bit more about that.
I then have the immense headache of keeping an eye on all of the material that goes out into the garden. From a point of view of record keeping, one of the other hats that I wear is DNA collection. I also do a lot of verification of plants that may only be known to genus level. So here you can see a bed that's by one of our ponds. I think we were probably at about 80% mapped when we moved into Iris, and I think we're now at about 95%. So it is actually fairly easy to find plant material. And below you can actually see a photograph of the particular bed that we're looking at here. Staff all have access to Flora on their phones when they're working in the garden, so frequently they'll update records while they're basically weeding beds or doing any maintenance work in there, which certainly makes finding things so much easier. It does cause me an immense headache every night because I sometimes have 20 to 30 sets of data arriving to be added from Flora into Iris each night, which just take quite an amount of time to upload in and then create a new data set. A slight new departure of all of this is the ability that we have now to track material that is in sites that actually aren't even in Edinburgh. The first one that we tried this with was one of our native plants, the alpine blue sow thistle, which is incredibly rare in Scotland. And we decided to experiment with tracking the actual plants that we put back into the wild. And this was smiled on very favourably by the conservationists that funded the work, because they have an immediate, very good record of where, in some instances, non-native plant material was put. This plant actually is so poor at regenerating that we've tried in some instances to make a population that contains some Norwegian DNA to try and give it some vigour so that it will actually reproduce in the wild. And they sat in different conservation sites. And behind two people who are planting the Cucurbita on a precipitous slope there, which you can see above them, you can then see the challenge that we had to actually try and map all of the plants on that particular slope. But it does mean that now that we have the ability to find them fairly easily. And our most recent departure with some of the plant material for the elms in particular that is going out at the moment, we're putting NFC tags buried just underneath them, which basically has got a good percentage of the data on. And we've built a little Android app which can then read the data from those NFC tags. So if labels are lost, we can find them, or if the plant disappears, we can still find where it was. We bought some quite expensive ones which have a range of about 3 feet, so that they're quite easy to find with an Android device. And I think the person that's done that has put a request into Iris to support that in the future. And that's basically the end of the presentation. If you want to interrogate our collection, you can use the barcodes down at the bottom there to get to the various different parts of our collection. And rather topically, we have a Garden Explorer page at the moment, which you're going to hear about in the next presentation. And unfortunately, you won't see any of the greenhouse plants on that, because our greenhouses are being completely refurbished at the moment and the public doesn't have access to them. So in that instance, you can use the Search the Living Collection plant search, which is built now out of Iris most recently.
Thank you so much, Ross and Peter. That was a great overview of what you do. And remember to sign up for the coffee chat next month on the Garden Explorer.